The final talk is being presented by Eric Verspoor. Eric has worked on population genetics and ecological genetics, particularly related to salmon, for many years. He was formerly with Marine Scotland and is now with the University of the Highlands and Islands. And Eric is going to, in 25 minutes, make us all experts in salmon genetics.
I wish. As I'm not an expert in salmon genetics, as you'll see, I can hardly make you an expert, but what I can do is make you aware of some of the basic genetic principles that are important that need to be taken into consideration. Genetics is a complicated science. It makes astrophysics look simple, to be truthful. This is going to be Genetics 101 for fisheries biologists. So don't expect anything. There's going to be a few complicated things in there, maybe, but the general messages are the important thing. What is genetics? Well, I'll tell you what it's not, and what a lot of people think, and not just your laymen, but also your scientists. They think that genetics is molecular markers. Well, Ken started his speech, and I thought, gee, he's giving a talk on genetics. And I thought, gosh, I hope he doesn't keep on with that, or I'll just have to stand down and not give my talk. Genetics is a pervasive issue in all living organisms. It deals with trait inheritance from parents to offspring in humans, in salmon, in mice, in dogs, whatever. It controls reproduction. That's what Ken was talking about. That's genetics. All biodiversity is fundamentally genetic diversity. That DNA variation underlies all the biodiversity that we see. It's genetics. It underlies domestication and selective improvement on salmon farms, cattle, dogs, whatever. Genetics. Wherever you look, there's genetics. And we now know, as a result of Watson and Crick in 1953, that DNA is the basis of that. That's the mechanism which transmits and the machinery of inheritance that underlies all this. So what we have to recognize at the base of every salmon, there is genetics, whether we like it or not. Whether we like it or not, gravity is there. Whether we like it or not, electromagnetism is there. So we have to take genetics into account. What we have to understand is what the implications of genetics are for what we do, and that's the tough ask. The fundamental divisions that we have, really, when we look at genetics are, there's the individual. That's the package in which DNA we see out there. It's packaged. We can define that. Everybody knows what an individual is, an individual salmon, an individual human being. The breeding population, we know what that is conceptually, but that's a very difficult thing to define. Where does it stop and where does it start in a river? Is there one breeding population? Is there many breeding populations? You can't go out there and canvass the salmon and ask them who they bred with and where their offspring are. So you have to find ways of actually interrogating the stock in a river to find out who bred with whom and who didn't, and how this is divided up into these breeding populations. And these populations in sexually reproducing organisms, if you have one salmon, it's no good. There won't be any salmon there the next generation. You have to have a breeding population, just like you do on farms, just like you do for perpetuating dog breeds or whatever. There has to be a group of breeders. It's the same in the wild. Then we have the species, and that's the outer boundary of it in a sense, in a good species anyway, because good species don't breed with other species. So that's the limits. Does a species have one breeding population? Does it have 100? Does it have 1,000? What? But those breeding populations are the units that Ken was talking about. They produce the next generation. They produce the excess that we want to harvest. These are the production units out there for your salmon stock, the breeding population. And population genetics is about the genetic processes that underlie that production of salmon. Now, genetics, like computers, is simple in concept. Mendel showed us that with his peas. There's some nice little rules in there. But like computers, which are based upon gates which are on and off, open and shut, and you look at your computers today and you tell me if you understand what's going on inside your computer. And that was built by humans. And there's very few people in the world that would understand the complexities of a computer that you're using. Well, we're dealing with genetics, and genetics is far more complicated. But it's based on very simple principles. But you ramp it up, and it becomes more complicated. I was quite interested in this here, the genetic realities. We have thousands of interacting functional DNA units in any individual. And these units are more or less shared amongst individuals in a breeding population. But if you look at the scale of what we're dealing, here's a fish scale, 1 centimeter or so in size. A cell, 100 micrometers in size. Chromosomes, 10 micrometers in length. That's after they've been stretched out at a particular phase of the cell cycle. And here, in 2012, somebody finally took a picture of DNA. And the size of this here is 20 nanometers. That's that there. And in here, if you look very closely at that, you can actually see the individual bases in the DNA. This is with an electron scanning microscope. For the first time, we're starting to actually see directly the world of DNA. And when you think that the DNA in a salmon, one individual salmon, if you took all that DNA, you could wrap it around the world several times. That's how long, what you've compressed into these chromosomes. Now that's incredibly complicated, but based on a very simple principle. Population genetics. Well, that was the individual there. Well, you're going to take lots of individuals. You've got lots of individuals which are interacting, interbreeding. It becomes even more complicated. Again, a male spawning with a female sounds very simple, but it's involving lots and lots of these genes that are interacting in lots of different ways. We have interactions between individuals within populations, interactions between populations. We have interactions of the DNA with the environment. We understand the basic concepts. The theory of population genetics has been around for a long time. What we don't understand is the detailed workings of it for a given species. Even man, where they spend billions and billions of dollars, we're only starting to understand what it is. And the salmon genome is twice as big as the human genome. So it's potentially twice as complicated. Keep that in mind. Population genetics. In order to understand what's going on, we come up with lots of formulas or simple ones like this. And this is just an excerpt from one paper of the tens of thousands of papers that have been done on the various aspects of the mathematical relationships amongst genes and how this operates under different conditions of selection, genetic drift, mutation, gene flow, etc., etc. This is it's just like computers. This is incredibly detailed, incredibly difficult. So, where do we get our understanding from? We get our understanding from 4 sources, from breeding studies. That's the traditional source. We get it from characterizing traits and success of parents and offspring and how these traits are inherited. We now know that virtually every trait that a salmon has a component that is inherited. It also has a component of its variation that is due to the environment that it grows up in, or it is in at some stage of its life cycle. We can also analyze DNA variation directly, and we can exploit that analysis for molecular markers, which tag fish. It's just like a physical tag. That's the way that we tend to use it nowadays a lot. CSI, salmon. We also use this mathematical modeling because we have to look at these relationships and what happens when you mix genes in different individuals in breeding populations in different ways, and get your head around all those things going on simultaneously is impossible unless you use mathematical approximations or you define very objectively these relationships. The key issues that are relevant to stocking in relation to genetics are genetic variation among individuals, within and among populations, genotype-environment interactions and adaptation, structuring of stocks into breeding populations, adaptive differentiation of populations. Those are the key areas of concern in relation to population genetics. This is the one graph which it'll take me a few minutes to explain. It's by no sense a comprehensive account of it, but it illustrates the complexity of salmon populations at a genetic level, or human populations for that matter. All individual salmon are genetically different. We can take that as a given assumption. All breeding populations are genetically different. We can take that as a given. We don't know what those differences necessarily mean, but we do know they are genetically different. And this is a particular example where if you just took 3 locations in the genome of Atlantic salmon, each of them which had a possibility of 2 different genetic variants at that location, you can take my word for it, get 27 different genotypes. Now, if you looked at 2 populations, black population and the hatched population, and you said they have exactly the same variants in those 2 populations, but the frequencies of those variants are different. For each variant in one population, it's 0.9, and in the other population, it's 0.1. Now, if you take that and you do the maths on it about the frequency of the genotypes that would be produced, i.e. the maternal variant plus the paternal variant, because all genotypes have 2 variants associated with them. Every individual has 2 variants. Then you can look at the frequencies that you would expect of these different genotypes. And what you see, even with that, where the populations share the same genetic variants, is that this genotype here, which is the most common in population 1, is 500,000 times more likely to occur in that population 1 than it is in population 2 here. And conversely, this genotype is 500,000 times more likely. Now, if you think of a finite salmon population of 10, 20, 30,000, the probability is the vast majority of the fish in population 1 and population 2 will be genetically different. That's 3 loci. There's tens of thousands of loci in Atlantic salmon populations that are variable, and a lot of them have a lot more than 2 variants associated with them. Some of them will have 60 variants associated with them. Therefore, we can take it as a given that they're different. Genotype-environment interaction, a nice fancy term, but what does it mean? It means the fitness and character of a population and its abundance are a function of the cumulative fitness of the individuals and the amount of available habitat, the environmental carrying capacity for the genotypes in the population. All salmon are the result of an interaction of their DNA and the environment. Genetics is integral to a salmon. You cannot have a salmon without genetics. And that defines their character traits, how well they survive, how well they leave offspring, that interaction. Therefore, genetics underpins everything. And differences in the DNA of individuals give rise to differences in their character, their fitness, their likelihood of surviving. Adaptation, fitness, abundance, and viability. What's the relationship? Here's a little cartoon. This is a takeoff. Carlos will sort of recognize this, but I did adapt it with apologies, Carlos. I thought a rabbit wasn't appropriate. Carlos has one in a joint publication in the book where he's put rabbits in here, but I have filched a few of the other things from there. The principle here is that you have an environment. An environment is constantly fluctuating. And what you have is the genetics is trying to keep up. It's adjusting itself through natural selection to pick the right genotypes for that particular environment. Those are the ones that survive. And the next generation is slightly genetically different, and it adjusts to the historical environmental conditions on the hope that the future environmental conditions will be similar. Well, it's not hope. That's a bit anthropomorphic. But that's what it does. That's the way it works. And the fitness is defined by the difference between the genetics of the population and the environment. There's a fitness deviation here. And if that fitness deviation is large, less salmon survive. So this immediately translates into the demographics here. So that environmental variation combined with that genetic tracking and how quickly those things happen have an influence on abundance and numbers here. Now, if the environment changes too much or is outside of the niche limits for that species, it just can't go genetically in that area, then you get a huge fitness differential, you'll depress the population, and it may even go extinct. Extinction vortex, if you want to call it. We can debate that later, David. But I think this extinction is possible. I mean, you put a dam in a river, you're changing the environment, you're putting it outside the niche of an anadromous Atlantic salmon. It is not going to be able to get up the river and spawn. Simple as that. Extinct. Stocking in some circumstances could cause those kinds of environmental changes. The other thing is genetic changes that occur if you change the genetics of a population, because remember, it takes genetics and environment to produce a salmon coming back. Well, if you change the genetics of that population, say you introduce North American Atlantic salmon with 56 chromosomes instead of 58, they're all the same, aren't they? No, they're not. And I'll show you an example as to why that's a problem in a moment. If you do that with stocking, if your stocking isn't well considered, appropriately designed, etc., you're going to have a problem. So you can't ignore that as a possibility. Now, structuring into breeding populations. Our understanding of breeding populations relates largely to the use of molecular markers which tell us things about whether two groups of fish that have been taken from different locations or different periods of time actually can be derived from the same randomly breeding population. If they're genetically different enough, that just makes it extremely unlikely. And using that particular technique, what we've been able to demonstrate basically is that North American and European Atlantic salmon are very different. The chromosome numbers are different. Just about every locus you look at that's variable, they're different. In fact, you can tell them apart with virtually 100% certainty with just two molecular markers, as John Gilby showed a number of years back. That's how different they are. We know also that there's regional differences within Ireland. Phil, I've stolen this from you as per usual. No, that's mine actually. I stole that one from me. This is one I stole from Phil. This is regional stock groups in Ireland. And here's the Moy system in Ireland where we've got clear evidence of breeding population structuring within rivers. Now, this is just a few examples. There's lots and lots of these examples to the extent that now we can be very confident that there's a lot going on. But one thing you have to understand is that these markers that we picked are arbitrary markers. And a lot of times you read that somebody says, oh, well, you didn't find any genetic differences, or there must not be any differences there. But here's an example from one of John Gilby and my papers that there's 4 loci here that we looked at. If you had just chosen 3 of them, you would have said there was no structuring in the North Esk River system. But then you pick this one, an adaptive locus, MEP2, and you get massive structuring that's totally inconsistent with different breeding population, or consistent with there being different breeding populations. You then pick microsatellite loci here, and you find no evidence again, though you did find it down here. You then pick these new types of loci, the single nucleotide polymorphisms, and bang, you pick up the structuring again. There are a lot of biases here. Remember, we're looking at less than a fraction of a fraction of a fraction of 1% of that huge genome of the salmon. So what we're seeing here is a very small part of that picture. So we know there's a lot of salmon stocks out there. And in fact, if you look across the species range, there's probably tens of thousands of different breeding populations altogether on both sides of the Atlantic. There's lots of them. And we don't know the boundaries yet. We know there are a lot of them there, and we haven't necessarily differentiated them or really understood how they're related to each other. Each river structuring will be unique and conditioned by history and contemporary factors. So in Newfoundland, you get hundreds of populations of salmon that do not go to sea Many of them could go to sea. They don't. Why? Because of something that happened 10,000 years ago or so. Because they were stopped from going to sea by sea temperatures. They landlocked, and now the sea temperatures are fine. They don't go to sea anymore. Adaptive differentiation of populations, this is the real key. This is the area about which we know the least. And I'm afraid I'm going to go give you some examples that I've probably shown half the people in this room at various stages, but we haven't advanced that significantly in there. But they still illustrate the point that where we look, we find adaptive differentiation amongst populations to varying degrees. And we look at a few traits in that genome. There may be hundreds and hundreds of traits that are adaptively different that we haven't looked at yet. Maybe there aren't. Lack of evidence isn't absence or evidence of an absence of adaptation. We have to be very careful if we don't have the information to jump to conclusions. So there's the one about Gyrodactylus salaris. We know that Baltic populations are generally more or less resistant to Gyrodactylus salaris, the scourge of the British Isles and Ireland. If we brought that in here, the populations would be devastated as they have been in Norway. That's genetic adaptation on a regional scale. We also know from Davy Stewart's work from the freshwater lab on the transplantation of fish from the upper Tummel system and the almond, where we have timing differences in both adults and smolts, that these timing differences are maintained to a degree when you transplant them into a common mid-altitude stream, the Bran. But what we see also is that that timing is conditioned both by genetics and the environment. And that's true probably of the vast majority of traits. There is this intermixing. Another study that Mike Donaghy and myself did on egg mortality in the Kyla-Sutherland system, where we showed that the eggs from the Oikel catchment were resistant to acid flushes, whereas the eggs from the Shin were not. That didn't make a difference in the Shin system because there weren't any acid flushes, but it certainly made a difference in the Oikel system where there are. So if you put Shin fish in, you stock them, you get very high levels of egg mortality. Adaptation and outbreeding depression. One of the better examples that we have specifically for Atlantic salmon, it comes from Phil's work on the interbreeding of farm salmon, artificial interbreeding of farm salmon with the wild Burrish stock, and where we see that when you actually cross the farm salmon into the wild stocks, the fitness of those fish, the hybrid intergressed individuals, are lower than of the wild fish. That's one form of outbreeding depression. We also have another source of outbreeding depression, and that's associated with something that's called co-adaptation. If you're a salmon in North America and you've evolved for the last 600,000 years independent of the salmon in Europe, you evolve a group of genes that work together well. And in Europe, you evolve a group of genes that work together well in a certain way. You mix those two things up, and you potentially have problems. And here's an example of a very obvious problem. We crossed Scottish fish from one of the main East Coast rivers with Canadian fish that are one of these little residence forms that could go to sea but don't. They're 12, 14 centimeters long, males and females. 50 meters from the sea, they don't go to sea. They're about the two most different salmon populations you could get. Produce hybrids, they survive fine in the hatchery, no problem at all. You back cross them, these hybrid males, to the Scottish or to the Canadian. These ones survive perfectly fine. These ones die 100%. This is a co-adaptive difference between these genomes that screws it up in an asymmetric way. Now, this is just one illustration of what goes on in a hatchery, a benign environment, as opposed to the wild, on a very obvious trait, which is just survival of the eggs. What about those more subtle effects? We don't know anything about them. Nobody studied them. Are they there? Maybe not. Maybe they are. Potentially they are. We also have adaptation and inbreeding depression. We talk a lot about inbreeding depression, and we know it occurs in cattle and in domesticated strains and everything, but boy, do they inbreed these things, and we see it in hip dysplasia in dogs and all sorts of other things. But we have very few examples of inbreeding depression in Atlantic salmon. And here's one example from Niels Riemann in Sweden a long time ago, in which he showed that the recapture frequencies of stocked salmon relative to the level of inbreeding, there was a general decline in the recapture rate. A lot of noise around that, but a general decline. How big a factor is this? We don't really know. It's very difficult to study the effects of inbreeding in wild populations. So again, ignorance. Now finally, just mention one thing here, and this is to do with a combination of adaptation and population structuring, and we haven't really studied this in Atlantic salmon, but it's been raised, and in sockeye salmon in Alaska, there's something that's now being called the portfolio effect. And that is that you have lots of populations in an area, in one river, which contribute to the overall river stock. And one stock does well in one year because that year it's the one that's adapted to that particular environment at that time. And then the next year it's another one. And collectively you get stability in the stock production because you have these different portfolios, these different stocks that you're got in there contributing to the overall stock. Now, what happens if you eliminate some of those breeding populations or you compromise them? Then overall stock is going to be compromised, going to be less stable, potentially less productive, fluctuate more. This is something we haven't even explored yet in Atlantic salmon. Probably exists at least to some degree. Current state of knowledge in relation to salmon. I put the iceberg up there because really we've just touched on the tip of the iceberg. I've just kind of given you a little bit of the shape of that iceberg up there and what it looks like, and the fact there's a big part of it underwater and what that kind of shape might be. We really don't know a lot of the detail. And for that reason, when it comes to stocking, genetics is the elephant in the room. We can't see it. We know it's there, but we don't know what it really means for us. But that doesn't mean we shouldn't be thinking about it because it is the elephant in the room. So this isn't to say you shouldn't stock. It's to say just take this on board. Think about it carefully, what it might mean. Whether it might be a part of the problem that you have. Whether it might cause you a problem if you're going to do stocking. And like gravity or magnetism in the physical world, genetic issues are unavoidable. Genetics is integral to life. It is integral to salmon stocks, etc. Thank you.
Thank you very much, Eric. Spot on time. We have about 20 minutes before you get your lunch. So let's start first. Are there any specific questions to Eric? And then we'll get the three speakers together and we'll have a more general discussion, bringing together all their... Yes.
Beg your pardon?
Indigenous stock, where you have a river, you take about 50 fish a year, and yes, you breed from them, and you try and mix them up as best you can. What impact do you think that would have?
I think every situation is unique, and I try to use analogy. Well, let's go to the computer analogy. Would you sit here and could you discuss with a computer specialist what the problem with your computer would be? You would ask the specialist to actually look at your computer and suggest to you what the solution might be. Each computer would be different. Each computer will have unique problems. I've seen computers which look identical on the outside, which won't run the same software. And the engineer won't be able to tell me why, other than to suggest that there's something inside that's different. For me to give you a standard prescriptive answer would be inappropriate. You have to investigate and analyze your specific situation and take it into account. As you can see, what we don't know is much, much greater than what we do know. So there is no answer. Yes, it is probably better generally to use an indigenous fish if you're stocking it. But if you take your indigenous fish out of a river and put it through the hatchery and then put it back in the river, and you've just removed 50% of the spawning biomass and it's below the conservation limits, you may just have depressed the production of the river even further than you would have had you left it alone. Equally, in another system, you may have actually helped it. So I think really it takes very careful consideration of each situation.
David.
Yeah.
David.
That last point you made, you said you may even have helped it. Can you envision many situations where some sort of intervention will beat nature? For example, if you're down to a few individuals in the population, can you afford to let them to spawn inefficiently, ineffectively? Typically in small populations you find that many of the par are from one or two matings and many of the others have made little contribution.
Do you find that? Sometimes you do, sometimes you don't.
Okay.
Very interesting, Caroline Thompson did a study on the west coast of Scotland and found that genetic diversity in the small rivers on the west coast was just as high as it was in the large rivers.
Right. I think it was work actually on the Gurnett Burn.
Yes, Alan Youngson did that. He wasn't sure what would happen, and he didn't want to risk the possibility. But in the Gurnett Burn, there's an interesting situation, which is that one of the prime spawning areas also happens to be one of the crap spawning areas. And that is because of groundwater upwellings, which are associated with a bedrock ridge. So you get deoxygenated water coming up. And okay, in a good year when the salmon are distributed throughout the system, this isn't really an issue if 50% of them die because of this. But if you've only got 3 or 4 fish and they're all attracted to that one location, it could be a problem. So again, my argument is there's no generalities here. In evaluate each situation on its own merits.
There are situations in which you could suggest that intervention, taking the fish and doing some carefully designed crossings, could beat nature.
Yes, in some situations, yes. But again, which situations those are, I'm certainly not knowledgeable enough to be able to say that, and I don't think anybody is, to be honest. I think we suck it and see adaptive management.
So can I throw in then that if the angler or angling club out there is thinking of a stocking program, how do they go about evaluating some of these things? I mean, I'm not disagreeing with what you're saying. I'm just saying
If you're an angling club or a district fisheries board or whatever, and you're thinking about stocking, go back one step and think about what your objective is. I think it goes back to what David was saying. Yes. Is stocking the thing that is going to achieve your objective? If you want the ranga, fine. Science isn't going to tell you what the decision you should make. That's the decision you make. What science is going to do is inform your decision about the potential consequences that you may have as a result of your actions, or if you don't take actions. And I think my message is that we don't understand what the consequences in very many situations, what the consequences of our actions are. We don't think about it hard enough. We just go, it can't hurt.
But is your message then that we should be precautionary, that you shouldn't do this? I'm still unclear about how you get that information to decide whether you should.
If your objective is to maintain a natural, healthy, self-sustaining salmon population or set of populations in a river, yes, be precautionary. If your concern is to maximize fish to the rod, it's a different
matter. Okay.
Different question altogether.
Okay, before the people up here take over the conversation, anybody else have any questions to all 3 speakers? perhaps I could throw in another one. There's been a lot of talk about, particularly from David and Ken, about trying to establish what the problems are before you start. And Eric went back to this as well. And I guess, again, my question is, for a lot of the people in the room, they're going to say, well, how do I do that? What do I need to do? What's your general, sort of generalized advice? Or is it, you're not clever enough to do this, leave it to the experts?
No, I don't know what the answer is. I don't think it, to stand up here and say for an organism that we know very little about the genetics and very little about how the genetics interacts with the environment to affect trait characteristics, survival, fitness, etc. I mean, look at human medicine. We're just as variable. We know now that so many diseases have a genetic element associated with them. We also know that whether those genetic predispositions to get particular diseases are very much influenced by the environment. Cancer is a good example. If you don't go out in the sun, you're less likely to get skin cancer, even if you're an identical twin. So there's an interaction there. But different identical twins will behave differently because some of them will have genes which predispose them to cancer if they are in the right environment. It's very complicated. Would we as laymen consider that we would know the best way forward to manage all these conditions in humans? I don't think we can say that. So I think we do have a challenge, and that is managing in the absence of complete understanding and complete information. And I don't think that's something I alone can answer. I think that's something that this meeting really needs to consider. How do you consider what the best way forward is, and what the role of stalking is in the absence of the required understanding?
Okay.
That's a question to you.
Ken.
I should really have made mention of it. There's an excellent book that was produced last year by the IFM from Simon McKelvey and from Ronald Campbell, and I stole some of the bits and pieces out of that for my talk. And that actually gives you a very good recipe in terms of how you might go about this, because I think that's really what Ted was asking. And I think people should be encouraged to actually look at their own information before they jump in and before they start thinking about the solutions. And that's what I was attempting to do in the context of the sequence I was bringing you down through the eggs and so on. And I have this idea that if people think of it as a wheel, and think about the bits, the components of that wheel, the spokes, if you like, that you have a handle on as a manager or an angling club. So the most important thing you have is the catch. And if you can really, really get people to report that catch in an accurate way, it gives you incredibly useful information, but it needs to be associated with effort. Having got the catch and the effort, and you know then, you can take from the literature some idea of the number of eggs per female that might be involved in that particular stock. Again, you can take from the literature the potential ratio that might be there of males to females, depending on whether you have a grilse or whether you want to see winter stock. I think intuitively you can begin to build up a picture of how your fishery is doing, and you can then ask the questions of the experts in specific terms, rather than a situation where you feel you have to go to the experts to give you the information. Very often that information sits with you. And if your club is progressive enough to have taken a census of juveniles in your river, and most importantly, if you have some idea of the actual productive area of the spawning areas of that river, again, you could fill out different spokes of that wheel. So that's really what we're attempting to do with our little model system that we hopefully will have available to you in the next 6 to 9 months, is to get people not to produce an answer for their particular river in our case, but to get you used to the idea that you can gather your own information and you can begin to understand better where those pinch points are in the context of your own particular fishery. But I do think that we could make much better use of the information that's available to us if it's better structured in terms of the way the information is collected.
Yeah, I mean, adding to that thing on that model, of course, one of the key problems is identifying where those pinch points are, and that's really the question I guess I'm trying to get to, is how the man on the riverbank does that. We as scientists spend our life trying to do it, and there are a lot of challenges. Eric, quickly, and then.
Well, I mean, I sort of build a little bit on what Ken says. I think we need to build partnerships. One of the things is is for us as experts in our particular areas is to disseminate understanding and knowledge. The more people that have a basic understanding of things like genetics or demographics or whatever, the more likely they are to get to the right solution, the more likely they are to be sensitive to the issues that are going on. The other aspect of it is to understand that the only way that you, given in a situation where you have imperfect knowledge, is to accumulate knowledge through monitoring and assessment. Don't just do stocking without finding some way of evaluating whether that stocking is actually achieving the objective that you had in mind. And that way you can learn. And if we collectively, just like the RSPB, they join together people inputting information from various different programs and monitoring in the gardens together, and they collectively understand an awful lot more about what's going on with the birds in the UK, we really need to set up our own little studies with our own little rivers and make sure we monitor them properly in an objective way where we can learn. And the next time around, we can adjust what we're doing in the right direction, change tack, put it into the pool of general understanding, and then we'll get there and we'll get a much better understanding of the way to move forward. Even if we don't know the details of it, we may still know when stocking works, how the best way to stock is, etc.
Thank you. Jamie? Sorry, you've been gazumped.
Hi, Simon, it's my last foundation. Question for David. One of the arguments that's also commonly reported for hatcheries at the top is the idea of accelerating recovery, which is one of the things you did, which mostly comes from the experience on mine. In the case, one of the things that I'm trying to get my head around is with that argument, what is the risk of genetic damage to your running?
Right. I think in situations like time where the natural stock is never completely lost and where many of the problems facing the fish, which was predominantly water quality in the lowermost reaches and estuary, have been removed, then there is a very strong case for leaving things alone and letting them get on with it. What happened in the case of the Keelan hatchery and the time really clouds the issue. We don't even know to what extent the hatchery actually was involved. There were big arguments about it. But an investigation done by Nigel Milner a few years ago suggested that the contribution of the hatchery was not like as big as the pro-hatchery people claim. What the real impact of that hatchery has been on time, we don't know, and we may never know. Perhaps it could have been monitored, more marking down, more investigation, more genetic studies. It appeared to have got away with it. Time is now very healthy stock. So I would argue in situations like that where things are generally looking good and there is a local stock, the best thing you can do is to look after that stock, that environment, and let the fish do the rest.
Thank you. Jamie?
Thank you. Jamie Stevens, Exeter. So a comment on some work that's recently been done in my lab, and something that Eric might like to come back on. We've been working with two hatcheries in Devon on the Tamar and the X, and we've worked with them and analysed fish that have come out of the kind of routine crossing procedures that they undertake at those hatcheries, and the best of any of the products of those hatcheries have only been able to match up to diversity by a range of measures that we can study that we might achieve in the wild. Quite frequently, the worst of those protocols have resulted in a level of genetic diversity in the offspring that falls way below what we would see in the wild. So I think there's a real danger in the hatchery process, which, as Eric rightly says, can be very variable. It needs to be properly evaluated. There's a great danger that the very action of the hatchery process can lead to the fish that are produced being having much reduced diversity than we would see in the wild. And the good news in relation to the hatcheries that we've been working with is that they have taken some of these results on board and already looking at ways that they might
modify their crossings. That's not to say that they won't continue to stop, but they've taken that information on board. So,
okay, so at least they're being adaptive.
Yeah,
Eric,
one of the I absolutely agree with you, and that's just one of these on if you how much effort is it to get it perfect. But one of the things which has been pointed out by a number of people, of course, is mate choice, which is potentially incredibly crucial. We know that in many organisms, people choose each other on the basis, inadvertently, of their genetics. And that's related to at least one group of genes called the major histocompatibility genes, which are associated with the immune system, but are also interlinked with olfactory cues. And we know that that is likely to be a factor also in the case of fish. Now, if you go into a hatchery and you force two fish to mate, they may not be particularly compatible at particular sets of genes, and you're going to reduce the fitness of that mating. How do you decide that? Do you just look at this one group of genes? Will that tell you everything? No. But if you let the fish decide for itself, then it has it's the best judge of who its best mate is likely to be. So you can't overcome that particular obstacle terribly easily unless you set up large tanks in your hatchery, you put your fish in, and you wait for them to say, who are they going to mate with, and then you gather the eggs up at that point. I think we're starting to get into the uneconomic kind of scenario. But I think Carlos could comment even more on that particular issue than I can. Carlos Garcia de Llanes over at the table there, who's studied these kinds of things as well. But these kinds of issues, again, I can only raise them. They are there. They're an elephant in the room. Make no bones about it.
Yeah. Can I just pick up on one point there, Eric? When I was involved many years ago in trying to specify hatchery programs, I learned not to eventually. I always thought if you had, say, a dozen of each sex available, the thing to do is to divide the eggs and the milk into 12 to 144 lots, do every possible cross you could. Yet the general advice now is one-on-one mating, which they don't generally do in wild, of course. I mean, male salmon will reproduce with several females often.
Right. I'm not sure who says the general advice is one-on-one.
So you divide each fish's product to use with three others, or?
It's between
right. Why not 12 males and 12 females?
You can do that. I would say that's the minimum recommended. You can do any factorial exactly weight, so the more factorial processes you have, the benefit starts to tail off the normal of it. You could use 12 by 12, you should if you want to be really good, but we recognise that that is quite a considerable effort and we need quite a lot of the similar on. With the shad, for example, that we're doing in Wales,
Final question, Ian.
In Melbourne Environment Agency. Just going back to the point Jamie made about hatcheries not being as efficient as replicating the wild situation, and Ken alluded to it before, how important do you think precocious male par are in genetic diversity?
Well, given that they're an involved component of the populations, and in most populations they are major contributors, I think they're important. I think if you exclude them, you reduce the effective population size, which is from an evolutionary point of view, is very important. You reduce diversity. We don't know what their role is. I mean, we think because they're small, they're insignificant, but that's just our anthropomorphic sense that anything that's bigger is better. You see, a multi-sea winter fish is better than a grilse, isn't it? Well, is it? If you didn't have mixed strategy in terms of returning and that, you wouldn't have as stable a population. In Newfoundland, you go and something like 90% of the grilse or 90% of the fish in the rivers there tend to be grilse or more, even 100% in many rivers. And 90% of the males are precocious males. So if you didn't have precocious males, you didn't use them, you'd be in serious trouble there. So again, you got to work with your own river. Think very, very carefully about the specific circumstances. And, these guidelines of using 3 or 5, these are pragmatic things that say, yes, we're moving in the right direction. Don't set up a supportive breeding program without at least 50 males or 50 females. But we've only got 49 males and 49 females. I guess we can't do it. That's, if you have to take pragmatism in with these principles and integrate them into the best possible program for your circumstances, and then adaptively manage and monitor that and evolve, recognizing you're ignorant and you're starting.
Okay, thank you very much. And in the interest of keeping the programme on target for the rest of the day, and because I've been up since 4:30 this morning, we'll break now. I think, perhaps we could just have a quick thank you to all our speakers for providing an excellent introduction.